Good morning, students. Today we will discuss seed dormancy. Dormancy is a condition where seed do not germinate even when the environmental conditions such as water, temperature, and air are favorable for germination. In some fruit plants such as mango, citrus, the seed germinate immediately after extraction from the fruit under favorable conditions of moisture, temperature, and aeration. But in case of apple, pear, cherry, seed do not germinate even under the favorable conditions are applied. This phenomenon is called as seed dormancy. Dormancy is due to several factors. Next is type of seed dormancy: physical, physiological, combination, morphological, and morphophysiological. First one is exogenous dormancy. This dormancy is imposed by outward factors of the embryo. In exogenous dormancy, the tissue enclosing the embryo can affect germination by inhibiting water uptake, providing mechanical resistance to embryo expansion and radical emergence, modifying gases exchange, limit oxygen to embryo, preventing leaching of inhibitor from the embryo, and supplying inhibition to the embryo. It is of three types. Number one is physical dormancy. Seed coat or seed covering may become hard, fibrous during dehydration and ripening. As a result, they become impermeable to water and gases, which prevent the physiological processes initiating germination. This type of germination is very common in true fruits. Example, peach, plum, apricot, cherry, walnut, etc. Next is mechanical dormancy. In some fruits, seed covering restricts radical growth, resulting in dormancy of seeds. Some seed covering structures such as shells of walnut, pits of some fruits, and stones of olive are too strong to allow the dormant embryo to expand during germination. The water may be absorbed, but the difficulty rises in the cementing materials as in walnut. Germination in such seed does not occur until and unless the seed covering are softened either by cementing moisture and warm conditions during storage or by microbial activity. Next is chemical dormancy. In seeds of some fruit, chemical that accumulate in fruit and seed covering tissue during development and remain with the seed after harvest. It is quite common in fleshy fruits or fruits whose seed remains in juice, such as citrus, stone fruit, pear, peach, and tomato. Some of the substances associated with the inhibitor are various phenol, abscisic acids, etc. These substances are strongly inhibit seed germination. Next is endogenous dormancy. This type of dormancy is imposed by rudimentary or undeveloped embryo at the time of ripening or maturity. This can be of different types such as morphological, physiological, double dormancy, and secondary dormancy. Morphological dormancy. Dormancy occurs in some seeds in which the embryo is not fully developed at the time of seed germination. Such seeds do not germinate if planted immediately after harvest. Plants with rudimentary embryo produce seed with little more than a pro-embryo embedded with a massive endosperm at the time of fruit maturation. Formation of rudimentary embryo is quite common in various plant families. Some plants of temperate zone like holly or snowberry have rudimentary embryo. Next is physiological dormancy. Non-physiological dormancy. After ripening, time is required for seed in dry storage to lose dormancy. Temperate fruit crops such as apple, pear, cherry, plum, apricot, vegetable, and flower crop have this type of physiological dormancy, which may last for 1 to 6 months and disappear with dry storage. Next is photodormancy. Seed that either require light or dark condition to germinate are termed as photodormant. It is due to photochemically reactive pigment called photochrome widely present in some plants. Next is thermodormancy. Some seeds have specific temperature requirement for their germination, otherwise they remain dormant. Such seeds are called as thermodormant. For example, seeds of lettuce, celery, pansy do not germinate if the temperature is below 25 degrees Celsius. Physiological dormancy is of three types. Double dormancy. In some species, seeds have dormancy due to hard seed coat and dormant embryo. Such seeds require 2 years for breaking out dormancy in nature. Combination of two or more type of dormancy is known as double dormancy. Next is secondary dormancy. Secondary dormancy is due to germination conditions. The conditions can include high or low temperature, prolonged darkness, and water stress. It is of two types. Thermodormancy. It is due to high temperature induced dormancy. Conditional dormancy, change in ability to germinate related to time of year. Methods of breaking seed dormancy. Number one is softening seed coat and other seed covering. It helps in better absorption of water and gases, which ultimately lead to germination of the seed. This can be achieved by scarification. Scarification is the process of breaking, scratching, mechanically altering or softening the seed covering to make permeable to water and gases. Three types of treatment are commonly used as scarification treatment. These include mechanical, chemical, and hot water treatment. Number one is mechanical scarification. It is simple and effective method of scarification. Chipping hard seed coat by rubbing with sandpaper or cracking with hammer are simple methods useful for small amount of relatively large seed. Scarification should not proceed to point at which the seeds are injured and inner part of seed are exposed. Chemical scarification. Dry seeds are placed in container and covered with concentrated sulfuric acid in the ratio of 1 part of seed to 2 part of acid. The container should be of glass, wood, non-metal, or plastic. The acid-treated seeds can either be planted immediately when wet or dried and stored for later planting. Large seeds of some legume species, brinjal, tomato, are reported to respond simple sulfuric acid treatment. Hot water scarification. Hot water scarification. Drop the seed in hot water with the temperature ranging from 77 to 100 degrees centigrade. The seed soaked in water for 12 to 24 hours. The unswollen seed may be separated from the unswollen seeds by suitable screens. The seed should be sown immediately after hot water treatment. Next is warm moist scarification. The seeds are placed in moist warm media for many eons to soften seed coat and other seed covering through microbial activity. The treatment is highly beneficial in seeds having double seed dormancy. The seed fruit includes cherry, plum, apricot, peach, show increased germination if planted early in the summer. Next is stratification. Stratification is a method of handling dormant seed in which the imbibed seeds are subject to period of chilling to after ripe the embryo in alternate layer of sand soil for a specific period. It is also known as moist chilling. The seed can be sown immediately after stratification in the field. Seeds with a hard endocarp such as prunus, apricot, peach show increased germination if planted early in the summer. Next is outdoor stratification. If refrigerator storage facility are not available, outdoor stratification can be done either by storing seed in open field condition, in deep pits, or in raised beds enclosed or wooden frames. However, it is likely that seeds are destroyed outdoor by excessive rain, drying, or by rodents. The top is covered with sphagnum moss to maintain moisture level. The pit or tray is irrigated at regular intervals to maintain appropriate moisture status. Next is seed treated with growth regulators or chemicals. If the endogenous dormancy is due to the presence of inhibitor, we can apply growth regulator at the low level to break dormancy. Like GA, cytokinin, kinetin can be used at the concentration of 100,000 ppm to break dormancy. GA is a light substituting chemical. KNO3 2% can be used to break dormancy of light required seeds. Thiazura can be used for breaking dormancy for both light and chilling treatment required seeds. Ethyl can be used for breaking dormancy of cotton seeds. Nitrogen compounds like thiourea, nitric acid, nitrate can also be used breaking seed dormancy. With this, we have completed the seed dormancy topic. Thank you.